So first of all, thank you very much to the organizer for the invitation. It's a pleasure to be here. And thank you very much to all you that are interested in DeepEMhancer. So many people doing cryo-EM methods, we tend to think on the pipeline, on the cryo-EM pipeline as the process that goes from movies to reconstructed volumes. Now, it is true that you can tell many things about the protein from the volumes. In order to make the most, in order to answer the most interesting questions, you need to have models. And model building process is not trivial, and it is very difficult to obtain models directly from the refined maps. Generally speaking, you need to post-process the maps right before the model building process. Why? Well, there are at least two reasons. First of all, there is noise in your maps, and perhaps it is well less noisy than the particles, but still, when you are looking at low resolution regions in your map, it is sometimes difficult to distinguish noise from signal. And also, it is also well known that maps exhibit loss of contrast at high frequencies. And this is the kind of thing that is generally deal with sharpening approaches. And there are many different sharpening. Okay, I'm sorry for that. I don't know if I can do anything. So there are many different sharpening approaches. And among all of them, we are very interested in this locus scale approach, which is a local approach. That means that it deals well with heterogeneity. And it's also a knowledge-based approach that basically takes atomic models in order to post-process your map. And what it means is that if you have a high-quality atomic model for your map, then you just can use this atomic model in order to get a high-quality post-process map. But of course, if you already have the atomic model, well, probably you don't need to do the cryo-EM thing before. So this is one of the limitations, although it is true that you can try to use some homologous models or things like that. But nevertheless, this is an important limitation of this current approach, despite the quality that you can get from it. So what we tried to do in DeepEMhancer was just to get results of similar quality to this kind of post-processing, but without the requirement of any atomic model. And in order to do so, we need to get another source of information, another source of biases. And in this case are the structural databases, the PDB and the ENDB, from which we can get atomic models and associated volumes. So this is the source of training data for the methods. And this is how the actual implementation looks like. From pairs of experimental maps and atomic models, we employ this log scale to post-process the maps, and we also compute typed masks from the atomic models. And applying the log scale post-processing plus the masks gives you a very high quality post-process maps that they look quite close to perfection in many cases. So in order to replace that complicated part of the pipeline, we just train a neural network that is trained in such a way that, it tries to minimize the differences between the output and the ideal post-process map. And the good thing about this approach is that all these atomic model requirement, post-processing, masking steps, we have already done it for the training, and we do not need to care. The neural network has learned how to reproduce this kind of post-processing effect just using as input the raw maps. And this is the power of the approach. It has learned how to do a very complex post-processing or something very similar to it, just using as input the raw maps. And just to give you a couple of details, from a deep learning perspective, this is not a very complicated model. This is a classical 3D unit. We have tried different losses. In the end, L1 in real space was the best, but there were not many differences with respect losses. And the only thing is that since the cryo maps tend to be very large, we need to process them in chunks using sliding windows approach, as you see in the cat picture. And we cannot afford to have like large batch sizes, so we need to switch from typical batch normalization to other normalization techniques as group normalization. But apart from that, this is a very simple deep learning model. And even though we tried way more complicated models, in the end, the results were not too different. And we prefer this simple approach because it is way faster than more complicated models. And what it is very important is the data we use for training. And conceptually, it's simple. You just need experimental maps and you need post-process maps, high quality post-process maps. But if you go to the databases, there is a lot of variability, and there is a lot of details, such as the coordinate origins of the maps and the PDBs. So it took a lot of time to collect a set of EMDB entries and PDB entries that were of high quality and that they contained all the metadata we needed to be sure about the quality. And once we collect that data set, yeah. Once we collect the data set, the only two tricks that are common sense, if you wish, is that since we want to distinguish protein features from noise features, we need to standardize the size of the maps. So we always wanted to present them to the network at the constant size. One residue, one alanine should always look the same. And we also needed to normalize the voxels, the intensity of the voxels, because different reconstructions may have different scales. So we end up selecting this normalization technique, which basically just scales the noise to have 0 mean and 0.1 standard deviation. Something pretty typical. And although it's true that you can do other sorts of normalization, we got better results applying this strategy. And by the way, we use half maps during the training just to avoid EMDB entries that had as main map the post-process maps. There is no requirement to use half maps, but we didn't want to have already post-process maps as part of our training set. So this is the reason we use half maps in the training process. And well, this is one figure from the paper. Generally speaking, you can see that DeepEMhancer tends to improve the quality of especially the more problematic parts of your maps, like these distal parts. And if you zoom into these regions, well, you can see that the density, overall speaking, it looks more continuous, and in many cases, it helps you to trace missing loops or even some problematic side chains. And after that, many people have used that. So you can, if you go to the literature, to people using it, you will find very interesting examples. These are just a couple of examples in which you can see on the right-hand side how DeepEMhancer helped you to separate two strands that look fused in the first example, whereas in the second example, you can see how it helped to modeling some side chains that were not very clear at the beginning. I very like this paper because this is one of the first papers that use DeepEMhancer, and probably during the review process, since it was very, very new, well, they need to perform several, let's call them soft validation strategies. So I very like this paper in which, well, they show in this plot that the pink line, which is the sharpened map, tends to correlate poor. The correlation is lower for the sharpened map compared to the atomic model that when you use DeepEMhancer for the most part of the protein. And the second thing I like about that paper is that, well, they showed that if you use a global B-factor correction approach, in many problematic regions, you get broken densities. You get discontinuous and very noisy parts, whereas the DeepEMhancer map tend to be quite smooth and they are more easily interpretable. But what they do as a sort of self-validation, they showed that if you spend your time trying to pick a particular B factor for every particular residue, for every particular loop, then you can get results that are somehow quite similar to the ones that you get in DeepEMhancer. So what it tells you is that, well, the density were there, and basically this is just a sort of it's a matter of trying to find which is the best manner of filtering the results if you wish. But DeepEMhancer does it automatically for you, or at least it does among other things. But to be honest, this is the most interesting result from my viewpoint, is that I have just taken this from Twitter. DeepEMhancer seems to attenuate anisotropy, which is quite surprising. And well, visually you can see here that this map seems to be, well, the anisotropy seems to diminish after DeepEMhancer usage. And from the class distribution, from the particle angular assignment distribution, you can see that the map is indeed quite, it suffers from orientation, for preferential orientations. So yeah, that's something very interesting. And well, we have just checked in many other cases, and this is the famous hemagglutinin case. And yeah, indeed, it looks like it is dealing with this anisotropy. So we have tried to study this in a more systematic way. So we have computed directional FSCs comparing experimental maps and DeepEMhancer process maps against the reference PDBs for a bunch of examples. You have here a couple of those examples. And these plots, what they show is the FSC, directional FSC. The solid line is the mean value, and the shadow region is the one standard deviation of the directional FSC. So the narrower, the better. And you can see that, well, for an important range of resolutions, DeepEMhancer tends to exhibit way less anisotropy. So indeed, this visual effect that we can see, it is supported by those calculations. So we can affirm that, at least in many cases, DeepEMhancer is able to attenuate anisotropy, which is an unexpected result. And it's surprisingly good, let's be honest. But what it is even more surprising is that DeepEMhancer is not the only technique that can attenuate this anisotropy. We have checked other post-processing strategies, log scale, log spiral, and well, even though they are not able to do this sort of high quality anisotropy reduction, they are consistently better than the experimental map. As you can see here in this summary, this is the standard deviation of the directional FSC. So the lower, the better. And you can see that the blue line, that is the experimental one, it has a way more standard deviation for a very important range of resolution, from 5 to the reported resolution, basically 5 angstroms to reported resolution. And you can see that log scale, log spiral, and DeepEMhancer, they exhibit less deviation. Thus, they are less anisotropous. And this is one example using log spiral. You can see visually also that you get some correction on the anisotropy. So this is also another unexpected result. And it took us a bit of thinking, try to understand why it is happening. And well, let's say that the way that log spiral works is basically by normalizing the amplitudes of your map over the different frequency by frequency over the different directions. So this sort of normalization tends to homogenize the amplitudes around the directions. And well, this kind of thing could help. I guess that this is one of the reasons it does, but it is less clear. But for the case of log scale, it seems a bit more simple, since log scale make use of the atomic model in order to do the correction. So if you have an atomic model that by definition is not anisotropic, then it should be able to correct partially this anisotropy. But still, what is even more surprising to me is that DeepEMhancer, that in principle was trained to mimic this log scale behavior, it is able to do better than the log scale. So there should be something else that explains that. And well, to be honest, we don't have a good explanation, but we have a hypothesis that we try to prove. And we have talked a lot about embeddings, latent spaces, and we think that it is related to that. The way that DeepEMhancer works, well, it's a unit, but conceptually, you also have a sort of encoder, then you have a latent space representation, and from the latent space representation, you basically sample the output. And if you think carefully, when we train DeepEMhancer, we only use high quality maps. So when the DeepEMhancer that has only been trained on high quality maps is presented an anisotropic map, well, the encoder part would do its best, try to map in this input to some point in this latent space. And depending on how bad the input is, well, the result would be better or worse. But the decoder only knows how to translate latent space point into isotropic results. So by construction and by dataset generation, the decoder in principle, it's not able to generate anisotropic results. And this is our hypothesis. We need to do a few tests just to try to nail down this explanation, but we are pretty convinced that this is at least partially one of the reasons we see this anisotropic reduction. And lastly, I just want to comment on a couple of limitations about DeepEMhancer that you all should be aware of. It has only been trained on protein and nucleic acid data. So if you are facing ligands or you want to, or you have a strong translational modifications, it is likely that the results won't be that good. And many times what it happens is that those ligands or those translational modification, they go masked out basically. And the second thing is that, and it's related, some very low quality parts of your map, they can just directly be masked out also. But in some cases, you may be interested in preserving those. So for those cases, well, DeepEMhancer is probably not your best solution. And I would say just that since there are many other methods, then just try to pick other methods, not for the backbone, not for the side chains, but to focus only on the problematic regions of your map. And they probably would give you the most if you just try to combine different approaches. And that's all. I just want to thank all the people that were involved in this development, Javier Vargas and many people in the biocomputing unit, especially Carlos Oscar Zorzano, and also to my current supervisors in Oxford, Frank Bundelf and Charlotte Dean, and all the funding agencies. And thank you very much. I will be very happy to take questions.